And now I will hand over to Elana from Johns Hopkins for the last presentation here.
Hey, wonderful. Thank you so much for having me here today. I'm going to talk today about some computational methods that we've been developing for trying to study tumor heterogeneity, really scaling across different molecular data modalities with some recent applications to single cell, but thinking more generally as a computational framework of how do we really study transcriptional dysregulation in the context of inter and intra-tumor heterogeneity. So if we think about tumor heterogeneity and why this is an important problem, this brings us back, of course, to Leo Tolstoy, but really thinking about tumors are very much like unhappy families. And I love this quote for thinking about the algorithm contextualization of happy families are all alike, and every unhappy family is unhappy in its own way. And I think this is really true of tumor biology, where you have healthy tissue is really all behaving relatively homogeneous across different individuals in many contexts. But when you get to a disease, and particularly cancer, each cell and each tumor is really its own ecosystem that has very much dysregulated in order to give rise to the malignant phenotype. And the heterogeneity in cancer really occurs across disparate scales. So it occurs across patients, it occurs within a patient where you can have multiple tumors at different tissue sites that are related to metastasis. You could have, even within a single tumor and a single organ site, you can have multiple lesions that can have disparate profiles. And then there's also the cellular, genomic, and epigenomic states that give rise to further heterogeneity. And all of these play together to give rise to a particular phenotype in a tumor and its response to therapy or resistance. So one tumor type that I've studied a lot in this context that I think is a useful model system is head and neck tumors. And what's interesting about the head and neck tumors is that they both occur within the oral cavity region, but they have two very distinct etiologies, where there's one subset of the tumors that arises from tobacco and alcohol use, which gives rise to one set of phenotypes, and another set of tumors that arise from HPV infection in the same region, which again gives rise to a different set of alterations. And this makes them a very nice sort of ecosystem in which to study cancer biology and transcriptional heterogeneity, because you have these two subtypes that are naturally giving you sort of this biological experiment of heterogeneity. The other thing that's interesting about head and neck cancer is that in spite of their sort of pervasiveness and having several driver mutations, those driver mutations are really insufficient to explain the pervasive transcriptional dysregulation that's observed in these tumor types. So there's really more under the hood that really explains what are the phenotypes and what do you see in the disease that make them a useful system to study. So we're going to focus largely on the types of heterogeneity that we're able to study at the molecular and cellular level, where it's spanning across cellular, genomic, and epigenomic features. And it's fortunate in terms of a multi-omics approach that there's now measurement technologies that are really able to capture each of these, where the cellular states you're able to uncover from single cell RNA or CyTOF, the genomic from whole exome or whole genome sequencing, and the epigenetic from a whole slew of ChIP-seq, ATAC-seq, and other methylation and chromatin accessibility assays. And together, integrating these give you the functional outcome of the sources of variation through transcription. So now if we want to think about how are we going to quantify the type of heterogeneity that we see from all of these assays, we want to think about what's a corresponding computational metric that's going to give us that. And how do we sort of adapt this Anna Karenina principle of tumors to single cell or other transcriptional data sets to really get this robust statistic? Because most of our computational algorithms are focused on clustering or mean shifts through sort of differential expression that fail to capture this, where is something dissimilar versus where does it become more dissimilar? And so in order to do that, we developed a pathway metric called expression variation analysis, EVA, which was led by Bam and Afsari in my group, to compare the dissimilarity of transcriptional profiles from one group, let's say tumors in the blue group, to another group, let's say normals in red. And so how do we quantify where in this case you can see something that's very dysregulated in the red group, but very homogeneous in the blue? How do we start to develop this in a multivariate metric? So what Bauman realized we could do is we could use a dissimilarity matrix where we calculate for every single expression profile, what is the pairwise dissimilarity between groups of samples in the blue group and the pairwise dissimilarity between pairs of samples in the red group, and then formulate an estimation on the expected dissimilarity between every pair and ask what is the variation between them? How often is that expected dissimilarity smaller between an average pair in the blue group and an average pair in the red group? And it turns out you can use U-theory statistics in order to get an asymptotic estimation of the p-value for dysregulation in this context. And the nice thing about using a dissimilarity metric is then that can be very generalizable to a lot of different types of assays. And really the whole story comes down to what is the dissimilarity that you use in order to quantify differences between your multivariate profile. So the statistic that we get is going to depend very much on which metric that you adapt. So for a lot of our work, we've been focused on using the Kendall-Tau dissimilarity metric. So what is this? Why not just use a correlation or Euclidean distance? What the Kendall-Tau dissimilarity does is it takes a relative ranking between all your pairs. So let's say you have two profiles, Ni and Nj, representing two different normal samples. And here you have your 4 genes, and here are their expression profiles. What Kendall-Tau does is it basically does a bubble sort distance. And so it asks for every single pair of genes, how often is, if I take, for example, AC is smaller than AB in sample I, then I'm going to ask in sample J, does that distance preserve between AC and AB? Do I still see the relative dissimilarity matching up? And then count up the number of times that you see matching pairwise comparisons. So for example, this AB, this 14 is greater than 5, but 10 is less than 13 in NJ. So for NI, it's greater than, for NJ, it's less than, so you end up with one disagreeing comparison. The advantage of doing this is that it's comparing the similarity of rank orderings. So it becomes independent of the expression levels for each sample. So this metric can become robust to data normalization in the measurement profile because you're not depending on a variant, you're sort of having a within sample normalization. So this has been implemented along with EVA to quantify the dissimilarity between groups in the R package GSREG. So to go back and sort of test our initial hypothesis that led us to develop this, we wanted to see indeed are pathways more variable in tumors than they are in normal samples. So we took RNA-seq data from the TCGA and we quantified in head and neck tumors how often we took all the pathways in MSigDB, applied EVA with this Kendall-Tau dissimilarity, and asked how often are the pathways dysregulated with a Q value less than 0.05. And what we observe is that if we look at and then plot the variation in normal samples relative to the variation in tumors. And what we observe is that the statistics in all these pathways are shifted above the line y equals x, suggesting that they have in general higher variation in head and neck cancer. And indeed, more of the statistically significant pathways with greater dysregulation are occurring in tumors relative to normal samples, which sort of gives us proof of principle that it's, sort of, we're seeing this variation that we're hypothesizing to occur in tumors. So I talked earlier about head and neck tumors not really being genetically driven. So the question is, what is regulating that type of dysregulation that we see? And one of the large components and contributors to head and neck tumors in general, and cancers in general, but largely head and neck, are epigenetic alterations as a dominant mechanism for dysregulation of transcription. And so you can think about open chromatin, right? If you have a change in chromatin state that leads to more open chromatin, you have a tendency for transcription factors to be able to bind, and you're going to see more transcription there. Whereas if you have condensed chromatin that's relatively tight and packed with silenced transcription, you're not going to be able to have those transcription factors binding there as much. And so you're going to end up with less binding and less expression. So what we wanted to do is extend our model to see how much do these transcriptional alterations that we're seeing really contribute to the types of transcriptional heterogeneity that we're seeing. So if you see a change in chromatin state, do you see a corresponding change in transcriptional heterogeneity, where maybe our hypothesis is that it's not that open chromatin means that a transcription factor is going to bind and you're going to have enhanced expression. What open chromatin we hypothesize means is that you're going to have an ability for transcription to occur if there's a secondary transcription factor that binds. So what you're going to see is more heterogeneity in the transcriptional states rather than more of a mean shift. So what we did is take our framework that we developed in EVA for pathway dysregulation, but now apply it to gene sets where you can define sets of genes based on ChIP-seq or other chromatin, ATAC-seq, other chromatin accessibility data to compare and define a gene set of which genes are specifically open in normal relative to which genes that are specifically open in tumor, and use that in order to quantify your differential heterogeneity. So in the context of head and neck tumor, we had a data set where we had matched ChIP-seq data from primary tumors, our HPV positive, as well as normals and cell lines. And we used those ChIP-seq peaks to find regions that were open chromatin specifically in the normal samples, which I've shown in black, as well as regions that are specifically open in the tumor samples, which I'm showing in blue, and also showing in purple if they also occurred in HPV positive cell lines as tumor related. And what we observe is that in the normal state, where you have regions of open chromatin, what we plotted here is the regions of open chromatin, and then this heat map is corresponding gene expression data, which contains a match of a couple of these samples, as well as a much larger cohort of head and neck tumors, of 50 tumors. So we have 4 tumors with our ChIP-seq data, and now 50 where we're getting gene expression data. What we observe is that in the normal regions that are associated with chromatin state, with open chromatin in normal, we observe increased expression as well as more homogeneous expression in the normal samples, which are shown in gray. Whereas in the regions of open chromatin, we observe greater shifts in transcriptional dysregulation, where we observe increased expression, but with far more heterogeneity than we observe in the normal samples, sort of validating this hypothesis. And we can apply the EVA framework in this Kendall-Tau model in order to quantify this dysregulation that's arising from the open chromatin. So as opposed to it having a mean shift where we're constantly thinking about what's overexpressed, we're hypothesizing that we want to think about these changes of variation as the dominant features that are being driven from epigenetic regulation. So of course, this is all, nice in bulk data, thinking about chromatin, but we're now fortunate to be in an era where we have single cell data, which really provides us with new insights into heterogeneity of biological systems, that we can study each cell and its heterogeneity and its transcriptional state. And typically the way people are doing this, right, is, we draw our UMAP plots, we make our clusters, and we go, oh, well, there's 10 clusters here, therefore this group is more heterogeneous than the other. But we're never actually drilling down to the molecular states to say which pathways are more heterogeneous and what does that imply about the biology of the system. And this is particularly important in the context of this patient-level heterogeneity, where maybe you have single-cell data that's capturing multiple lesions from an individual, where you can start to evaluate what are the molecular pathways that are stable across lesions versus what are the molecular pathways that are really sort of unique to one, and how much of this arises from heterogeneity and a wide state of states in the initial tumor, suggesting that if we have a cocktail of therapies that are designed to block that original tumor and its metastatic mechanisms, could we target it better? Versus how much of those develop at a later stage of evolutionary history. So we took, in order to start thinking about this framework and what's going on in terms of metastasis and understanding the role of this heterogeneity in the context of metastasis, we reanalyzed public domain single-cell data of head and neck tumors, which specifically performed matched single-cell sequencing of primary tumors with lymph node metastases in the same individual. And in that original data set in Pyram et al., what they did is they applied the usual sort of clustering style approach to the tumor cells in the sample. And interestingly, what they concluded from clustering analysis and sort of a t-SNE style analysis is that what they observed is that the primary tumor cells and the tumor cells from the lymph node metastases clustered on top of each other. So there didn't appear to be transcriptional changes within the tumor cells, and they attributed a lot of the metastasis to the microenvironment. And we wanted to see, is that because of the reliance of this clustering style approach, or is that a feature of the data? And so we went through and applied our EVA framework in order to quantify pathway dysregulation. And now each dot here represents a hallmark pathway from MSigDB, where we've colored it in red for the metastatic sites, and in blue is our EVA statistic, this variation statistic for the primary tumor. And what you can see is there's a couple cases where indeed, yes, the variation appears almost identical between the primary tumor and the metastasis. But in 3 of them, we observe this shift where the primary tumor appears to have more transcriptional heterogeneity in these pathways than does the metastasis, which is consistent with this model of clonal evolutions being selected from a larger, more heterogeneous primary tumor that then solidifies in the metastasis. And now, as we're emerging from single cell technologies into spatial molecular technologies like Visium, we're able not only to get the metastasis and the tumor cells from within dissociated cells from the individual lesion, we're actually able to now map them on top of an H&E, which lets us look in an individual tumor at the multiple lesions and actually trace what's an invasive carcinoma from what are the pre-malignant lesions that will later form subsequent malignancies in that individual. And you can apply pattern detection methods like our own COGAPS approach to these data. And you can look at them and observe very clearly these different pre-malignant lesions are using distinct molecular pathways from the invasive carcinoma, suggesting that they're all having these different mechanisms that are occurring within the tumor. We can apply our EVA approach to this same Visium spatial transcriptomics data. And when we do that, what we observe is that the tumor cells themselves are more heterogeneous in terms of pathways that are regulating cell cycle and epithelial to mesenchymal transition. So we have a wide array of cells exploring that, cells rapidly dividing, giving this heterogeneity in the cell cycle in the primary tumor. And then when we start to look at the pre-malignant lesions, at least for a subset of them, we start to observe more heterogeneity in immune response, suggesting that there might be more immune attack of some of those specific lesions, which could also be confounding from the multiple cells captured in Visium. But there does seem to be an interaction between the tumor and immune cells. as well as the estrogen signaling pathway in this tumor, it then becomes more heterogeneous, consistent with our pattern detection algorithm that we're observing different hormone receptor pathways being turned on in the different lesions. And this really gives us rise now to think not only what are the different molecular pathways that are dysregulated, but then how do they occur not just at a pathway level, but how are they mediated by interactions through multiple cell types? For example, here, overlaying patterns that are associated with the invasive carcinoma with patterns that are associated with immune cells. And so now the next question becomes, how do you modify these dysregulation statistics in order to consider multiple cell populations that are interacting? So to summarize our work, EVA is a really robust statistical framework for heterogeneity analysis to compare variation between populations. Transcriptional heterogeneity really spans population, regulatory, and intra-tumor heterogeneity. And the EVA framework is very well suited to multi-omics, single-cell, and spatial data modalities to map inter and intra-tumor heterogeneity. And with that, I'd like to thank the members of my lab, particularly Bahman Afsari, who's now working in industry as a machine learning expert, as well as our collaborators. Bahman really led the development of EVA that's been picked up on in a lot of our subsequent applications to single cell and spatial technologies. So thank you, and I'm happy to take any questions.
Thank you very much for a very nice talk, Elana. So I can ask the first question. So you looked at this sort of heterogeneity of the epigenetic landscape in terms of these histone modifications, but you were sort of focusing on the genes, right? So can you also look at sort of distal regulatory elements and see if you have a heterogeneity there that can be quantified?
We could. We didn't see it as cleanly if we looked outside of the specific sort of promoter regions from the TSS in this case. I think we probably need another model than just the gene set level approach, because the problem is when you expand it out, your gene set's going to get huge. And when your gene set gets huge, you're going to see more variation just because you have more genes in it. And so I don't know how to sort of discriminate. We've been thinking about this, like, do you get sort of do you modify the framework or modify the dissimilarity metric to use like a kernel around it or something like that? You know, I don't know what the right approach is for that, but it's something that I think we need to think about a little bit. Yeah, but it's certainly, I think the same approach could apply, right? And maybe it's just a variation test instead of a mean test at a distance. But I do think this concept of variation as opposed to mean for transcriptional regulation is an interesting model.
Yes, very much so. Just trying to see here if there are any other questions. It does not look like there are any other questions coming up here now. So in that case, I will ask you another question. So with this whole single cell approach, do you think that, because now we have these sort of various multi-omics methods sort of coming online. So how do you think that will sort of benefit this analysis that you presented at the end?
Yeah, so I think for multi-omics, right, the obvious one is the combined ChIP-seq and ATAC-seq and single cell RNA, as well as DNA methylation, right? Like there's that, SCNMT-seq. So the obvious one is what's the regulation, and sort of this that we applied in ChIP-seq and RNA-seq, where we were doing it at a tumor level, we were doing it, we didn't have the specific cell type. So I think we have a much more ability to refine and ask questions about gene regulation through these sort of approaches. The challenge is, of course, the sparsity of single cell epigenetic data. So I think that's something that we need to address as we think about this and defining these regions. But I do think that that'll be an interesting area. And in the context of these spatial, technologies, I think that it's, we're really at the start, right? Like, and I think this ability to map what is the heterogeneity? Where is it going? Maybe incorporate things like copy number on top of it. How does that relate? And what's the clonal outgrowth that we can really, for the first time, I think, actually visually see and map in tumor systems is really exciting to me.
I could not agree more. I hope that we'll have another one of these seminars in a year or two, and then we can invite you to talk about those results. So I think we've reached the end of the line here for this symposium. So I'd like to thank the organizers, Yao and Vijay, as well as the people at the Center for Mathematical Sciences for pulling all of the administrative strings to make this happen. I'd like to thank all of the speakers who have presented their exciting work here today, and of course everyone in the audience who has been participating and helped make this a very successful event. So, as has been mentioned before in the chat for those of you who haven't seen it, there will be recordings will be coming up here on the symposium website later in the week. So then you can watch it all in slow-mo to see all the highlights and really enjoy them. So with that, I will put these proceedings to a close. Thank you very much.